All right, most of you have answered. And if you answered variegated, you were right. So you picked up on the cues that I said, this is her idea and this is how she explained the variegated corn. So she saw variegated corn and she came up with the ideas of the pieces that jump around. And in reality, it's that the transposon that breaks a gene, it could be breaking the genes in some cells, and then in other cells it didn't break that gene, and that's how you would also have these different colors, right? Instead of like jumping out, but it can jump out of the gene. So in this case, you would get variegated color. What is the other point? The thing that is interesting about this is that this is happening during mitosis, which is when the cells are dividing. So when we think mitosis, we think the new cell will be exactly the same as the previous cell, right? Unless there is some mutation. Many things lead to mutation. One of the things that leads to mutation is the jumping of transposons. It's not that the gametes changed, right? Because the different gametes are formed different meiosis. But when you look at the one kernel, all the cells should have been the same because the cells are all being divided through mitosis and they all have the same initial DNA. But this is to show to you that during cell division, DNA is not static, it's dynamic. It can be changing, and part of the change is mutations. Maybe it's too much sunlight and you get a bone in DNA that leads to skin cancer, or it's because of transposons. So just two examples. Hope you found this story very interesting, because this opened the idea to, wow, there's what else is there out there about transposons? So people think about transposons as jumping genes. You may call them transposable elements. So sometimes in molecular biology, if you see you're going to see the word element, and sometimes you're going to see the word factor. Whenever we say element, then it's referring to DNA. So like when I say regulatory element, so it's some DNA that's regulating something. So it's really regulating transcription when people talk about regulatory element. So element is DNA, factor is protein. When somebody abbreviates it as TE, transposable elements, it's the shorthand for both transposons and retrotransposons. These guys have viral ancestry. That's how you end up with them. And this one has different types of ending up in our genome. And let's see, they move around the genome, and I already told you a lot about her. Hopefully you're as amazed by her as I am. So prokaryotes have transposable elements, but eukaryotes have a lot more. So when I said have an idea of constitution, what I want to tell me, or if I show you two different pie charts and I ask you, which one of these two do you think is prokaryote and which one is the eukaryote? What I'm hoping you gather is that if there is a bunch of transposons or transposable elements, whether it's retro or transposons, then it's an eukaryote. So we have half of our genome is those guys. In corn, most of it, which is very upsetting when people were sequencing corn, they're like, I wanted to know which genes make corn, but you know, you're spending all this money sequencing DNA and it's always these transposons. But oh well, fact of life, we learned that corn has a lot of transposons, a lot of retrotransposons. And these guys are responsible for plant genome enlargement. Okay, so out of this slide, what do you need to know? You need to know that prokaryotes don't have a lot of transposable elements, eukaryotes do. So do the transposons also jump in the genes? They may. Maybe they just jump into something that is not a gene, and there will be no change in phenotype. But if it goes not in the gene but in the promoter, then they can make the expression higher. So meaning, when I say overactive, it means there's more transcription of that gene, and it can lead to cancer. If they get inserted into the gene itself, then that leads to hemophilia. That's people know what causes hemophilia, and Duchenne muscle dystrophy. Anyway, that's another genetic disease that's caused by transposons. Sometimes people call them selfish DNA because we don't know what it is doing other than just perpetuating itself. And then we're like, oh, we hate transposons, but they can be beneficial. Here's one example where they're beneficial for bacteria. Suppose this orange thing here is a transposon. Okay, well, here is one bacterial cell. This is another bacterial cell. Bacterial cells sometimes have conjugation, which is kind of their version of having sex. So there's a pilus, a hair here, that allows transfer of DNA from one cell to another. Actually, if it's just plasmid, it may not even need the pilus. So they can absorb a plasmid from just the environment, can go in from that came out of some other cell. All right, so here's a transposon. This is the bacterial chromosome. So bacteria have chromosome plus this smaller circular DNA. Those are called plasmids. And the transposon may go to a plasmid, and the plasmid may be carrying resistance gene to antibiotic, for example, and then that gets transferred to another bacterial cell. So if the resistance gene was in the chromosome, then the transposon helped it jump to the plasmid and then go to another bacterial cell. So this has been observed. So that's something that is beneficial to bacterial cell. In our case, so we're like, okay, I don't care about bacterial cells, especially because it's causing me harm. Why would I want transposons in my DNA? Well, they say, okay, they are shuffling the exons, which increase diversity, which maybe will lead to a less fit organism or a more fit organism. You're just increasing the different versions of something you have. And if the version is better, it will stay in the population. If it's worse, then that individual won't reproduce or will not be fit enough to survive. The transposon is not making the decision. It's just shuffling the cards. It's increasing diversity. So there are many types of transposons, but mostly I want you to remember these: retrotransposons and DNA transposons. The retrotransposons, they transpose through a copy and paste mechanism. And the DNA transposons, they're cut and paste. What does that mean? The copy and paste means that the retrotransposon copies itself, and then that new copy inserts itself in the genome. And that happening many times leads to a genome increasing in size. That's part of why we believe plants have such large genomes, is because they have lots of retrotransposons. The DNA transposons, they are not, they don't make a new copy. It's the same card. Imagine that this is a deck of cards. That card just gets shuffled in the pile. Whereas in this one, you have a new card, mysteriously being inserted in there. So these are the types I wanted to remember. And you heard about the AC and the DS. Remember, the AC is the activator, and then the DS is the dissociator. Remember, I told you the DS is the child that doesn't move around unless the activator picks it up. That DS was a non-autonomous. That's the one Barbara McClintock saw. It doesn't encode what's called a transposase, so it needs somebody else to move it. So there are transposons that are non-autonomous and some that are autonomous. But mostly I want you to remember retro and DNA transposons. So this is even more saying there are retrotransposons, and some of them are non-long terminal repeats, and some of them are non-autonomous, and some are autonomous. This is more for if you go to graduate school, so I'm going to skip that, but it's still emphasizing that there are DNA transposons that are cut and paste, and there are retrotransposons which are copy and paste. And yeah, that's main goal of this slide is to emphasize that there are many types of transposons, but those are the two types I want you to remember. And the autonomous versus non-autonomous. The reason why I'm putting these words here is because in the last slide, last class, I pointed, oh, these are all transposons, and move on. So here they are appearing again. The alu is the most common transposable element in humans, and the line 1 is another one that is common. So yeah, so the autonomous and non-autonomous is it able to jump by itself, or does it need another transposon that will make a copy of it or carry it around? All right, so that one, it's a figure again about 2 different types. So you can see how people love classifying these. Then there's long terminal repeats that happens with viruses that integrated themselves into our genomes. And then there are these other types, and some autonomous, some non-autonomous. Okay, and how much you have of each type of retrotransposon depends if you are rice, if you're a human, or if you're Arabidopsis. So these mites are a type of transposon, LTR transposon, and non-LTR, they're all transposons. And so all of these are different types of transposons, and depending on what you are, you have a different amount. All right, let me get to the C-value paradox in the last 3 minutes. So the C-value is how much DNA you have in the haploid nucleus. Okay, so that's the amount. If you weigh out the DNA, that's how much DNA is in that nucleus. It correlates to the count of base pairs, but let's just emphasize that's how much DNA you have in the haploid nucleus. We used to think that the more complex the organism is, the larger is its genome. So far so good. You guys agree with that expectation?